Hi, I'm Katrina. I'm an MD PhD student at the Center for Heart and Innovation in the Francis Lab. So, atherosclerosis is, in the form of ischemic heart disease, the leading cause of death in the world today. Atherosclerosis occurs when cholesterol from lipoproteins in the bloodstream accumulates in the cells of the artery wall. These then become foam cells. Our lab has recently reported that the majority of foam cells making up plaque are derived from smooth muscle cells as opposed to macrophages, which is what was previously thought. Smooth muscle cells differ from macrophages in that they have an impaired ability to efflux cholesterol. Cholesterol efflux is absolutely required for the regression or reduction in size of atherosclerotic plaque. We've also shown that smooth muscle cells compared to macrophages have markedly reduced levels of lysosomal acid lipase, which is the only enzyme in the lysosome that can hydrolyze the cholesterol esters that these cells take up from the bloodstream. We've also shown that adding exogenous LAL to smooth muscle cell foam cells in culture enhances cholesterol efflux from these cells. And a collaborator of ours has shown that adding exogenous recombinant LAL intravenously to the bloodstream of a mouse model of atherosclerosis also causes regression of disease. So we want to take this therapeutic model and employ it in another mouse model of atherosclerosis, and eventually, hopefully, humans. But we need to find a way that is not quite as expensive as recombinant protein to sustain high levels of circulating lysosomal acid lipase in the blood. And so we're exploring the use of mRNA, delivering that in a targeted fashion to hepatocytes such that the liver can be used as a bioreactor to sustain high expression of LAL in the bloodstream. And we know that lipid nanoparticles are a great way to deliver things in a targeted way with low immunogenicity. And it's been shown that mRNA encapsulated in lipid nanoparticles can be used in mouse models to sustain high levels of circulation of target protein. Because the atherosclerosis model I'm using is an ApoE knockout, the best way to target to the liver would likely be with an isogalactosamine PEG lipid. And so that is the plan. This is new and interesting because to date, therapies for atherosclerosis focus on lowering lipid levels in the blood. They don't target smooth muscle cells, and they don't typically focus on regression of atherosclerosis, which is important for disease modification. So that's why this is interesting, and LNPs with mRNA have not yet been used in the field of athero. There are LNPs with siRNA that are being used to match on P69, but this is something new and exciting. So please ask me questions, and I'll talk about it later.
That was 2 minutes, 53 seconds. Well done. Time for one question from each of the judges. Are there any questions?
Yes. Has there ever been someone tried in vivo just to use maybe I missed it, the protein itself? Like not in vitro, but in vivo?
Yes, sorry, I didn't say it super clearly, but yeah, there has been in one mouse model of atherosclerosis, a collaborator of ours gave recombinant human LAL intravenously, and that caused regression of disease. So that's the only one so far, other than using LAL knockout models and adding it back in, but that doesn't really answer the question that I'm looking to answer. So.
Another question, anybody?
You mentioned the cost of recombinant proteins. This is maybe not a fair question, but do you know the relationship of cost between the recombinant protein compared to the mRNA?
I don't, but based on things I've read, it seems as if this is a good option. And I think it's neat too that we're looking to sustain high levels of expression over time, whereas with the recombinant protein, I would assume that those levels would drop off a little more quickly.
Agreed completely. You have a feel for, this is a proposal, of course. How much sustained expression would you expect over time with mRNA?
Right, so I know that it's been published that with weekly doses, we can sustain expression. It's been shown, I think, at least up to 6 months, if not more. And I think, I would assume that would be long enough to cause significant regression of atherosclerotic plaque, but of course we'll have to see.
Very good. Thank you. All right, next up we have Jerry Leung from the Castrop and Cullis labs.
Right. Hello, everyone. As was introduced, I'm Jerry. I am a second-year master's student, co-supervisor between the Cullis labs and the Castrop labs here at UBC. And my proposal I'll be presenting is on using lipid nanoparticles to create designer platelets. And so to begin with, some of you might be wondering, well, why platelets? And I'm sure as many of you are already aware of, platelets are the cells that are traditionally known to be important for many roles, most importantly being blood clotting, however in recent years being implicated in other functions such as immune modulation, as well as even cancer metastasis. Clinically speaking, platelets are used as platelet transfusions in order to supplement patients that are low on platelet counts, such as when patients have leukemia, they have low blood counts, including platelets, and therefore platelets are transfused in order to restore their hemostatic balance. One of the problems, however, with using platelet transfusions is that they're not always effective. Either in a prophylactic setting or in a treatment setting, platelets, they're not necessarily very effective in terms of restoring that hemostatic balance. And in addition, platelets cannot be stored for very long. Usually shelf life is about 5 days, 7 days we'll be pushing it, but after that they cannot be used any further. One of the potential solutions is to use enhanced platelets that have maybe increased function, maybe more hemostatic potential, or maybe a longer half-life. However, the problems that have been currently faced so far is that they're difficult to modify ex vivo, and it's very difficult to do so. My proposal, therefore, is to use LNPs in order to achieve this modification. Essentially, you would have a bag of platelets, bag of donor platelets, and take some LNPs here for illustrative purposes, including encapsulating mRNA, but the same principle could be used for siRNA. You could deliver these LNPs to platelets, which take them up and eventually express the protein of interest, if it is mRNA, or a certain protein will be knocked down in case of siRNA. And the potential here is that if successful, this can create a platform technology that can not only result in more effective transfusions, such as when you have more hemostatic proteins being produced in these platelets, but at the same time could be used for targeted therapeutics. For example, if you can make platelets express anti-cancer proteins, or it could be even simply used as a research tool to investigate different physiological processes depending on protein expression or knockdown in platelets. And that is everything I have to present. Thank you.
Well, what would be your first proposed target to go after to validate your proof of concept?
Well, in terms of a proof of concept setting, simply a protein such as GFP could work. I'm sure platelets don't express GFP in any way whatsoever, so you can simply just have the LNPs encapsulate the mRNA for GFP, transfect to platelets, and under, for example, flow cytometry, see whether or not you get some green platelets. And of course, if this works on a consistent basis, then you can move onwards to using mRNA encoding for more physiologically relevant proteins, such as thrombin or factor 7, for example, in the application for hemophilia. Right.
Another question?
My question is maybe related. How feasible do you think this is for large-scale production of like engineered platelets?
I imagine it'd be towards the more feasible side. Already, LNPs are being used in a clinic via Patro, so there's already the scale-up technology in order to scale up these LNPs. And at the same time, platelets are already being used for transfusion, and therefore there's already a system in place to obtain a large amount of donor platelets. And this really is just a case of a marriage between these two systems. Hopefully not too messy of a marriage, depending on how the process goes. But definitely, the technology is there, and it's just a matter of proving whether or not this is feasible, starting off with proof of concept experiments.
Very good.
Okay, thank you.
Thank you, Jerry. Okay, and last but certainly not least, we have Amy Strilchuk, also from the Castrop lab.
All right, great. Thanks for the introduction. So yeah, like Bob said, I'm also from the Castrop lab, so luckily you're already quite familiar with what we do after the talks today. And I'll be focusing again on using gene therapy delivered by lipid nanoparticle to treat coagulopathies or diseases of blood coagulation. So The goal of all of our work is hemostasis, and that is healthy clotting. So being able to form a clot in the cases that you need it, so in response to an injury, but not forming clots when they're not needed and when they become pathological, such as here on the top left, where they can cut off blood flow to certain tissues or even get dislodged and cause other problems like heart attacks and strokes and these kinds of things. So all of this focuses on maintaining clot stability. And there's kind of two handles to clot stability, and one is coagulation, which is where most of the drugs today focus. And this has yielded drugs like anticoagulants for thrombosis, which work very well at preventing thrombotic clots, but they work almost too well in that they prevent clots from forming even when they're needed. So someone that was at risk of thrombosis suddenly becomes towards a hemophiliac side, so at risk of large amounts of bleeding even after a minor injury. And then similarly, for hemophiliac patients, some of the bypassing agents that stimulate clotting give them a risk of thrombosis in nonspecific clotting. So we're focused on the other handle that controls clot stability, and that is fibrinolysis, named after the lysis of fibrin, which is the protein that makes up blood clots. So just a little schematic is shown in the bottom right of how the coagulation cascade controls the production of fibrin from its inactive fibrinogen, zymogen. And then plasminogen, activated to plasmin, is the enzyme that actually lyses fibrin clots and is responsible for degrading clots when they're not needed. So there are actually many protein factors that regulate these processes, both promoters and inhibitors of the fibrinolysis process, and they kind of target different areas in the system. So as you got an introduction to earlier, factor 13 cross-links fibrin and makes it actually resistant to lysis. And so this is one of the targets that we're focusing on, and one of the many targets that we can apply liver-delivered lipid nanoparticles to change their expression in the liver. Thanks.
Time for judges' questions.
How would you approach this, like from an experimental point of view, similar question as the other one, and say thinking about different siRNA constructs, like you would first screen them like in vitro and then?
Yeah, so like you've already seen, we've done some screening to find siRNAs that knock down these proteins. I think really we would start by thinking about what disease models we want to apply these to. And what patient populations we'd be aiming to serve. So in the case of thrombosis, there are many mouse models, so we can start with screening mouse sequences. And then for hemophilia, obviously the best model and the one that all hemophilia treatments go through is the canine hemophilia model, so we'd start there as well.
I have one more question, but I'm only allowed to ask one.
Could you comment on what you think the translation of successful delivery of the siRNAs to the liver might be to, say, other disease states?
Just other diseases besides coagulopathies?
Yes.
Or, I think there's a lot of potential. A lot of things are made in the liver. Either augmenting their expression using mRNA or silencing them is definitely a useful tool. I mean, blood diseases are very easy because almost all of the proteins in blood come from the liver because it's not itself an organ. So they're very readily targeted with the lipid nanoparticle technology we have available today. It might take some time to develop the LNPs that target the other organs that may, synthesize the proteins implicated in other diseases. But I think that the model itself has great promise.
Very good. Thank you.
All right, thanks.